metabolism and ATP energy, or adenosine triphosphate energy. So metabolism involves a couple of things. It involves catabolic reactions and anabolic reactions. Okay, um, all this stuff is required for your body to function, cells to function, um, and so catabolic reactions are energy producing, is what you need to remember there, and anabolic reactions are energy requiring, right? So a catabolic reaction producing energy, right? Energy is stored in the form of ATP. Whereas in an anabolic reaction, energy is required, right? And then energy is used by breaking off one of the phosphate groups from ATP, because ATP is adenosine triphosphate, right? And then when you break off a phosphate from there, it makes it adenosine diphosphate, ADP, and one phosphate. And it's all cycle, goes back and forth and stuff. So, but ATP is the cell's energy storage molecule, right? And the important thing there is that it links energy producing reactions with the energy requiring reactions of the cell. Okay. We won't dive too deep into it, but you still need to know some stuff. So there are stages of metabolism, right? There's the digestion and hydrolysis, degradation, and oxidation, 3 stages, right? And they're all three are pretty self-explanatory. Um, Digestion and hydrolysis occurs when you're eating either your proteins, your sugars, polysaccharides, or your lipids, right? And after that, you've gone through and done the mechanical work and chemical work in your digestive system to break down those particular biomolecules as far as you can until so they pass through the cell membrane, right, where they go into the second stage, where it's degradation and some oxidation to smaller molecules, where the cell membrane breaks it down, right? And so those proteins, sugars or polysaccharides, and fatty acids all go inside of the mitochondrion. Their broken down forms go into the mitochondrion, becoming acetyl-CoA, right? And then acetyl-CoA is the first step in going into the citric acid cycle, right? Where FADH, the electron carriers, FADH and NADH, are used in the electron transport chain to produce ATP, right? And that's stage 3. Okay. So, just a quick review there. In the 3 stages of catabolism, large molecules from food are digested and then degraded to give smaller molecules that can be oxidized to produce energy. Okay, so that's just a brief overview of the stages. So we're going to be talking about the cell, right? And so in an animal cell, these are the basic parts that you need to remember. You need to know the cell membrane, right? That's the external surface of the cell. So the molecules that you consume, the food molecules you consume, get broken down and get passed through the cell membrane. The cytosol is the liquid inside the cell. Consists of the cellular, it's the fluid. They say the fluid, but I call it the liquid. The liquid part of the cytoplasm that contains many enzymes and many cell for the many cell chemical reactions. The cytoplasm consists of the cellular components between the cell membrane and the nucleus, right? They're pretty much the same thing. I mean, I wouldn't make too big of it. They're different, right? There is a distinction there, but you don't really need to know the specific differences between a cytosol and a cytosol and a cytoplasm. Sorry, yeah. Just remember that the cytosol is the fluid part, right? So mitochondria, that's where we're going to focus today. It contains the structures for the synthesis of ATP and energy-releasing reactions. We're going to dive deep into the mitochondria a little later on in the lecture. And then the nucleus is where all the genetic information is done. It's kept, right? We saw that in the previous lecture, nucleic acids. And then the ribosome is where we do protein synthesis, right? There's the ribosome right there. With the amino acids and the mRNA and the tRNA, right? And quick review: what are ribosomes made of? They're mostly made of rRNA, ribosomal RNA, right, and some proteins. All right, so let's keep moving. It's kind of a long lecture, so I don't want to dwell too long on any one slide. Okay, so here's ATP, right? Adenosine triphosphate, right there. I think this is the first time that I've ever had it written out. Adenosine triphosphate. You can see here, adenosine triphosphate is the adenosine part, the same adenosine part as from DNA, right? Except it has 3 phosphate groups attached to it. Okay. And then adenosine triphosphate is, of course, 3 phosphates attached. Adenosine diphosphate, 2 attached, and adenosine monophosphate is 1 attached. Right. So you can store a lot of energy in ATP. So when you hydrolyze ATP and make it ADP with a phosphate, an inorganic phosphate floating around, you actually gain 7.3 kilocalories of energy, right, per mole. And then if you break down ADP, hydrolyze it again to make it AMP, adenosine monophosphate, and an inorganic phosphate, you get another 7.3 kilocalories of mole, kilocalories per mole of energy. Just so you're aware, I've been saying this, but just so you know, remember, whenever you have a P, that's a phosphate, and the I there just means inorganic phosphate, right? Because it's charged. When it's attached to the adenosine, then it's a part of that entire molecule. But when it's by itself like that, it's called an inorganic phosphate. So for short, PI for short. PI for short. Next. Oops, I went too far. Previous. There we go. So just real quickly here, let's talk about how we digest glucose, like in our digestive system, right? Remember, we can eat in our mouths, put in our mouths polysaccharides, things like bread, or rice, or things like that, potatoes, right? And they're all polysaccharides of like amylopectin or and things like that, right? They're all polysaccharides. Inside your mouth, there's salivary enzymes. One of them is called amylase, and amylase can be used to break down those polysaccharides into smaller molecules. Shorter polysaccharides like destrins, maltose, and singular glucose, right? Into the stomach it goes, right? And the dextrins get broken down even more in the small intestine into maltose, and then maltose breaks down into 2 glucoses, right? And then even lactose gets broken down into a galactose and a glucose in your small intestines with the lactase enzyme. And then sucrose, just plain table sugar, uses sucrase to break down into fructose and glucose, right? And then once all those polysaccharides are broken down into their monosaccharide forms, it goes into the bloodstream and it goes into the cells where we do glycolysis, right, and other stuff like storing it. But in this particular lecture, we're just going to talk about glycolysis and the steps after glycolysis. So now let's briefly talk about the digestion of fats. This will be the only time we talk about fats in this lecture because we won't have time to go through fatty acid metabolism. But remember, when you consume fats from like animals, like milk fat or butter or the fat from steak or bacon fat, it comes in the form of a triacylglycerol, right, or triacylglyceride. And that is basically 3 fatty acids attached to a single glycerol, right? Here, our fatty acid is palmitate, right? It's 16. And when you're consuming fatty acids or digesting them, it's the same process all the way up until it's the same process as glucose or polysaccharide digestion up until you get to the part of the small intestine, right? The difference here now is that you have lipases, and lipases will come down and break down the triacylglycerol into its substituent fatty acids, right? And then also a glycerol. So these, when they get broken down, they encounter the intestinal wall, where they get transformed into triacylglycerols and then turned into lipoproteins, right, in the intestinal wall. And remember, that was the chylomicrons. And then after that, that's when it goes into the lymphatic system. They go into those lipoproteins go into the lymphatic system, into the blood cell, and then into the cells for use, either for storage or for use as energy as their substituent fatty acids, right? And the fatty acids they have to be broken down from their triacylglycerols to pass through the cell wall, and for them to be used in fatty acid metabolism, which you can't see down here because my head is covering it, but this part right here just says glycerol plus fatty acids. Because a triacylglycerol can't go into fatty acid metabolism, right? Or fatty acid breakdown, like glycolysis and sugars, right? It's the same thing because polysaccharides can't go straight to glycolysis, and your body has to break them down into their monosaccharide forms, right? So same thing here with fatty acids. Before they can go into fatty acid breakdown and fatty acid metabolism to produce energy and oxidation, they have to be broken down into their substituent fatty acids in the cell before that can happen. And then next we have the digestion of proteins. Same thing again here. We won't be talking much more about protein metabolism. So just very briefly, pepsin is in the stomach, and it breaks down the proteins into smaller polypeptides. There, it passes through the small intestines, where chymotrypsin and trypsin break down to their substituent amino acids, which can then be passed through the cell wall, the bloodstream, and then to the cell for usage or storage, right? So that's all I need you to really know about digestion of proteins. So just a quick review about oxidation and reduction. And then in the metabolic pathways, because that's how we gain energy in the metabolic pathways, specifically in glycolysis, and then the TCA cycle, the citric acid cycle, and the electron transport chain. It's all a series of oxidation reduction reactions. And what you're doing, since we're focusing on glucose here, is you're oxidizing glucose, right? And that's what's going to give you all this energy. So oxidation was loss of hydrogen or a loss of electrons, right? And it increases the numbers of bonds to oxygen. In a reduction, you gain hydrogens and electrons, and it decreases the number of bonds to oxygen. So that's an easy way for you to be able to identify what happened in a reaction, right? If you had an oxygen a part of a molecule before the reaction, and if it either gained bonds or lost bonds to it, then it was either an oxidation or a reduction. You can tell the difference that way. Right, and this is just another chart saying the same thing. Loss of electrons, loss of hydrogens and oxidation, gain of oxygen, and a release of energy. That's the important part right there. When you do an oxidation, you get a release of energy. And then in reductions, you gain electrons, and then you gain hydrogens, right? You lose an oxygen, but this is an input of energy, or energy storage, right? Energy storage. So there's several different coenzymes that are involved in glucose metabolism, right? And one of the most important is NAD. NAD stands for nicotinamide adenine dinucleotide, right? And it's considered an electron carrier. Or in glycolysis, we use NAD to carry electrons. And how we do that is we'll have NAD, right, come in, and what it will do is it will oxidize a molecule, for example, ethyl alcohol here, with alcohol dehydrogenase. And when it oxidizes it, notice that we lose a hydrogen in ethanol here after we oxidize it, right? And then that hydrogen that was here, right, is now here on NADH. So NADH is the reduced form. It's carrying electrons right now. NAD plus is the oxidized form. It does not have any spare electrons that it just gained. And then just as a convention, we always write NADH plus H plus, a hydrogen, right? Just to keep it balanced. Right? Here are those structures. You don't need to know them outright, but here's an NAD plus right here, right? Here's an NAD plus here, and here is the NADH, and that hydrogen, right, that it got when it oxidized in this reaction, ethanol, goes right here on the very top of NADH, right, plus a hydrogen right there. We have another coenzyme that's also very important too, called FAD. FAD stands for flavin adenine dinucleotide, right? In a similar fashion, NAD can be used in conjunction with succinate dehydrogenase. By the way, you ever see an enzyme that has a dehydrogenase in it, it's always going to be an oxidation reduction enzyme. Okay. So FAD, right, can oxidize succinate right here, right, and we'll lose these two hydrogens, making a double bond right there, and we'll get FADH2. Notice it's FADH2, and since there's already two hydrogens there, you don't have to have the plus hydrogen like we did with NADH. Okay. We only see FADH though in the citric acid cycle, which we'll talk about later. Okay, and just so you can see the structure, we have the entire structure of FAD here, right? And the business end is right here. And this position and this position is where the two hydrogens get added, and you've gained electrons. So right now, FADH2 is storing energy in the form of electrons, right? One more coenzyme, coenzyme A. Coenzyme A is a big molecule, contains pantothenic acid, which is vitamin B5, phosphorylated ADP, and then aminoethanethiol, right? This is this big molecule right here, coenzyme A. Coenzyme A is going to be seen right at the very beginning of the citric acid cycle. Sorry, I got a notification right now, just now on my iPad. So coenzyme A is going to be seen right at the beginning of the citric acid cycle, and usually you'll see it as acetyl-CoA, right? And so same thing here. This is a coenzyme that's used in the citric acid cycle, but it's not. We don't call it an electron carrier. All you need to know is that it's going to be a part of the citric acid cycle, right? That's why you don't see a reaction over here on the side that shows it gaining electrons or anything like that. Okay, next. So glycolysis is actually two stages or two phases, two phases, and I shouldn't say steps because there's multiple steps in glycolysis, but it's in two phases. And those two phases are the energy investment phase and the energy generating phase. The first 5 steps are the energy investment stage, where you actually put in ATP and ADP, right? And then steps 6 through 10, right here, right, is where you get energy finally being produced in the form of NADH right here, right? This NADH will later go into the electron transport chain to produce energy. And then, so the switch from energy investment and energy generation, right, happens here at step 6, and we'll take a very close look at that in a second. And then glycolysis ends with pyruvate being made, and pyruvate goes into the pyruvate dehydrogenase enzyme, which is a very big complex enzyme. And then after that, it comes out it goes into the citric acid cycle. But let's go through and look at these steps very deeply now, or not super deeply. So I'm going to go through this, one by one, because there's a lot of steps here and we don't want to get confused. So in the first step, we have phosphorylation, okay? So you have a glucose here, okay? And the glucose, using hexokinase, the first enzyme in glycolysis, along with an ATP, right, will phosphorylate the glucose right here, right? And that becomes glucose 6-phosphate, right? Glucose 6-phosphate. And then in step 2, we have our glucose 6-phosphate encountering phosphoglucose isomerase, right? And what it does is that it turns glucose, which was a hexose, right? It turns it into a pentose. Right? A fructose now. Okay? So, and then in step 3, we have phosphorylation. So, in this phosphorylation occurs with the enzyme phosphofructokinase. Right? So, in the previous step, our fructose actually is fructose 6-phosphate because it has a phosphate already attached to the 6 carbon. Fructose 6-phosphate encounters phosphofructokinase along with an ATP and becomes fructose 1,6 biphosphate, which is self-explanatory. You have 2 phosphates. One is on the 6 carbon, one is on the 1 carbon, and that's why it's fructose 1,6 biphosphate now. Right? Then in step 4, adylase comes in, right, and a cleavage occurs, right? And you get 2 molecules, this molecule and this molecule. Make sure you can see it on the screen there. My head's not blocking anything. One is deoxyacetone phosphate, and the other one is glyceraldehyde 3-phosphate, right? Glyceraldehyde 3-phosphate. It literally splits the fructose in half, okay? Splits the fructose in half. But the thing to notice about it is that now both of them are 3 carbons long, right? 3 carbons long, and they both have a phosphate attached, right? Right there and right there. So triose phosphate isomerase comes in right next in reaction 5, isomerization, right? And turns the dihydroxyacetone phosphate into another glyceraldehyde 3-phosphate, okay? So now you have 2 of these. The first one that was made right here, right? And then the second one that was just made with triose phosphate isomerase right here. So just remember now that you have 2 of those. You have 2 G3Ps, right? 2 glyceraldehyde 3-phosphates. So you should already glean from the fact, from this reaction, the fact that from a single glucose molecule, right, we can get 2 molecules that can be oxidized to make energy, right? So, 1 glucose is worth twice as much energy, and you'll see what I'm talking about in a second. But just keep that in mind, right? We have 2 molecules now. Okay. So that ends the energy investment stage, right? And the energy investment happened in step 1. It happened in step. Where did it go? Right there. Sorry, too much red around. In step 3, right? We needed the ATP, right, to do the 2 phosphorylations. Okay? So keep that in mind. So then starts the energy generating phase. And then here we start with reaction 6, which is oxidation and phosphorylation, right? We'll take one of those glycerol 3-aldehydes here in step 6, and we'll use it with glyceraldehyde 3-phosphate dehydrogenase, right? Like we were talking about before, what do dehydrogenases do? They're oxidation reduction enzymes, right? And so In the presence of glycerol 3 phosphate dehydrogenase, right, we'll oxidize glycerol 3 phosphate, right? And in that process, the electrons that we took from glycerol aldehyde 3 phosphate go to the NAD plus, right? And when they go to the NAD plus, we make an NADH. And the NADH can go to the electron transport chain to make energy. After that, our product is 1,3-biphosphoglyceraldate, glycerate, right? Glycerate. And 1,3-phosphoglycer. Why is this one so hard for me to say? 1,3-biphosphate phosphoglycerate. That's why I'm saying it wrong. It's not phosphate glycerate. It's phosphoglycerate, right? Encounters the enzyme phosphoglycerate kinase. Okay. And when it encounters the enzyme phosphoglycerate kinase, it will phosphorylate or transfer a phosphate, right, to an ADP, making an ATP. So you get an ATP, an energy molecule there. And I forgot to say in the previous step, one of the phosphates from a free inorganic phosphate also gets phosphorylated right there on that oxygen, right? So in this reaction, it wasn't just NAD plus as a substrate with the glyceraldehyde, as a coenzyme with the glyceraldehyde 3-phosphate dehydrogenase. We also had a floating around inorganic phosphate too. And in the process of this reaction, we also phosphorylated this molecule. And that's why now it's 2 phosphates, 1, 3, right? Bis phospho, right? So remember that this particular reaction is an oxidation. This particular reaction is an oxidation, right? Along with, which is what made the NADH from the NAD+, along with the phosphorylation, which is what added on that extra phosphate group to make the 1,3-bisphosphoglycerate. Okay, moving on. So 1,3-bisphosphoglycerate, along with phosphoglycerate kinase, right, will transfer a phosphate group to ADP, making ATP, the one that it just got from above, right? And now you have 3-phosphoglycerate, right? Just 3-phosphoglycerate. And you get an ATP. So you're just making all kinds of energy here. You've got some NADH here that will go into the electron transport chain to make energy, and you have an ATP here to make energy now, right? So in step 8, 3-phosphoglycerate encounters phosphoglycerate mutase, right? And what happens then is that it actually changes the structure of the molecule a lot, right? So if you recall, this part right here, right, the phosphate group here was actually down here before, right? So it actually moved this entire thing up, right? It had switched these places. The OH is up here now. The OH is down here now, and the phosphate group has moved up to the second carbon, right? So it's exactly what it sounds like. It's a mutase, and it mutated that molecule. It mutated that molecule into 2-phosphoglycerate. Then we have an enzyme called enolase. It comes in and does a dehydration reaction, which is exactly what it sounds like. We lose a water, and when we lose a water, we come up with this molecule, phosphophenol pyruvate. Right, phosphophenol pyruvate. Right. The missing thing here is we had the OH there, and now it's gone there. Right. It's missing there. This phosphophenol pyruvate in reaction 10, right, gets another phosphate transfer where pyruvate kinase comes in and transfers a phosphate group it transfers a phosphate group from phosphophenol pyruvate to ADP, making another ATP, right? and we're about to end glycolysis, and we've already made 2 ATPs, right? 2 ATPs already, right? So that's why it's the energy generating phase. And the cool thing about that is that once we lose this phosphate group right here, right, that's gone now, we make the molecule pyruvate. And the pyruvate goes into the pyruvate dehydrogenase complex, right? And that ends glycolysis. So in the overall reaction of glycolysis, we started out with a glucose, right? We ended up with we started with a glucose, 2 NADHs, 2 NAD pluses, 2 ADPs, right? And we finish with 2 pyruvates, both of which can go into glycolysis, 2 NADHs, and 2 ATPs. Right? Some water and some protons too, but that's the important part right here. This is the energy per generation part right here, right? All these can the 2 NADHs go into the electron transport chain to make energy. The 2 ATPs are already energy, right? Energy storage molecules, and then the pyruvates will go through the citric acid cycle to make more NADH and an FAD, and then H, and then those will go into electron transport to make more ATP. So let's talk about some pyruvate pathways now. When we had glucose, it went through glycolysis and made us pyruvate, right? Well, pyruvate can take a couple of different paths, one of them of which is what we'll be talking about for the rest of the lecture, which is the aerobic path, right, into the mitochondria. But the other way, just briefly, it can take an anaerobic path too, and that occurs when you're oxygen depleted and you've been exercising and such like that. The anaerobic conditions cause the pyruvate to go straight into the cytosol of your skeletal muscle, right, where it can get reduced by an NADH, right, and it becomes lactate. And interestingly, a buildup of lactate, right, in your muscles is what causes it to become tired and sore, right? After exercise, a person breathes heavily to repay the oxygen debt to reform pyruvate in the liver, right? And that's why you're trying to catch your breath after oxygen, after you exercise. But we're going to focus mostly for the rest of this lecture on this part right here, where anaerobic conditions in the mitochondria. So this is a citric acid cycle, right? It's a pretty big cycle, which I blocked some of it here with my head, but that's okay. Because we're going to go through each step, not in too much detail, right? But in enough detail. In enough detail. So now let's jump into the citric acid cycle. But before we can do that, you need to notice that up here, we start with acetyl-CoA, right? And I told you before that before the citric acid cycle, at the end of glycolysis is pyruvate. Well, pyruvate actually goes into an enzyme complex called pyruvate dehydrogenase, right? Pyruvate DH. And in pyruvate dehydrogenase, pyruvate gets turned into acetyl-CoA, or acetyl coenzyme A. And then acetyl coenzyme A is what goes into the first step of the citric acid cycle, right? So there is a step in between the citric acid cycle and glycolysis called pyruvate dehydrogenase. I didn't put a slide in there for that because I don't want to focus on that enzyme too much, but it is a very important enzyme, and you do get NADHs from that complex, okay? Because it's a dehydrogenase, right? So it's going to be doing some oxidation reduction, right? So just remember that there is a pyruvate dehydrogenase in between glycolysis and the citric acid cycle. So as we look at the citric acid cycle, you can see that it's a pretty long cycle. It's 10 steps long, or sorry, it's 8 steps long. And part of my head is blocking part of it right here, but that's okay. Like I said, we're going to go through each one of those steps individually in a second here. But notice all the NADHs that are being made. There's one right there, there's one right there, and one right there, right? And there's also an FAD that's being made here at succinate dehydrogenase. So we get a lot of our electron carriers being made in the citric acid cycle, in the steps of the citric acid cycle, which means there will be a lot of oxidoreductases, right? Dehydrogenase enzymes. So let's go through each step one by one now. in the first step of the citric acid cycle, in reaction 1, we start out with acetyl-CoA, right, or coenzyme A, along with some water, and it becomes citrate. Okay, so that enzyme is called citrate synthase, right? Remember what I said before? Biochemists are very simple when they name things, and you just took acetyl-CoA here, and after you got acetyl-CoA, you put it into citrate synthase, and it made citrate, right? So literally, citrate synthase is an enzyme that synthesizes citrate, right? Citrate synthase. After that, in step 2, we have the enzyme aconitase, and aconitase does a isomerization of citrate into the molecule isocitrate, right? So this is isocitrate right here, okay? And the difference between citrate and isocitrate is the position of this hydroxyl right here, right? That hydroxyl, okay? Isocitrate continues on into isocitrate dehydrogenase, where it gets oxidized, producing NADH and H+, of course, right? Our first electron carrier to go to the electron transport chain from the citric acid cycle. And at the same time, a CO2 is produced. And if you didn't already know, this CO2 right here being produced, along with a couple others in this cycle, are the CO2 that you breathe out when you breathe out CO2, carbon dioxide, right? I had this interesting situation once when I was at a FedEx store, and I was shipping some mass spec samples to the University of Georgia, I think it was, and they were on dry ice, which is frozen carbon dioxide. And I thought it was not a big deal because I used to get Omaha steaks from Nebraska, right? Shipped to me on FedEx, using FedEx on dry ice. So I was like, okay, this is easy. Let's just put the samples on the dry ice, keep them frozen, ship them via FedEx. Well, little did I know that the person at FedEx knew very little about what CO2 was, because she thought that frozen carbon dioxide was dangerous. I tried to tell the person that they were breathing out carbon dioxide, and they proceeded to tell me that I did not know that. And in fact, I did know that. It is them that did not know that they were breathing out carbon dioxide. So yes, this is the same carbon dioxide that you're breathing out. It is the same carbon dioxide that you see here in this step of the citric acid cycle, isocitrate dehydrogenase, right? Step 3. And so since you lose this carbon dioxide right here and you breathe it out, this step is both an oxidation and a decarboxylation, right? Because you oxidized isocitrate to become alpha-ketoglutarate, but in the process you also decarboxylated and lost an oxygen, right? And you also remember, just to reiterate, we get our first NADH from the citric acid cycle. Alpha-ketoglutarate goes into alpha-ketoglutarate dehydrogenase. I don't know if you're starting to see a pattern here, but the substrate is always in the name, and what happens to it is also in the name of the enzyme. So alpha-ketoglutarate dehydrogenase does what? It oxidizes alpha-ketoglutarate, turning it into succinyl-CoA. But in the process, we get another decarboxylation, right? And a second NADH being produced from the oxidation of alpha-ketoglutarate. Okay? So from succinyl-CoA, right, we go into the 5th reaction, right? And in this 5th reaction, we get hydrolysis, right? And this reaction is performed by the enzyme succinyl-CoA synthetase, right? And succinyl-CoA synthetase turns succinyl-CoA into succinate, right? All it really is doing is that it's removing the coenzyme A, and in the process, we get a GDP and an inorganic phosphate having a phosphate transfer to become GTP. And then GTP is very similar to ATP in that it's an energy carrier, right? And GTP can actually become ATP once it encounters an ADP, right? In adenosine diphosphate. Okay. The next enzyme at step 6 is succinate dehydrogenase, right? Again, and I don't know why there's not the name here. Oh, it's right here. And just like the name says, succinate dehydrogenase comes in and oxidizes succinate, making it fumarate. And the way it does it is that it uses an FAD, the coenzyme FAD that we talked about before, turning it into an FADH2, right? Now, succinate dehydrogenase is actually very special because it also serves as complex 2 in the electron transport chain. And so, previous steps of the citric acid cycle where you were making an NADH, those were being transferred from the citric acid cycle to the first complex of the electron transport chain, which we'll see in a second, right? But succinate dehydrogenase is actually a part of the citric acid cycle, but it's also known as complex 2 of the electron transport chain. So that FADH2 that's being made by succinate dehydrogenase from the succinate, right, is its direct connection to the electron transport chain, right? The TCA cycle has a direct connection to the electron transport chain, where one of the enzymes, succinate dehydrogenase, is actually also complex 2 of the electron transport chain. That's important to know. Remember that. So succinate becomes fumarate in step 6, right, in this oxidation reaction. And then moving on to step 7, fumarate gets hydrated. Okay? Fumarate gets hydrated. And somehow something got cut off there, but that's an H2O. Okay? And fumarate gets hydrated by an enzyme called fumarase, and it becomes malate in step 7. Okay? It becomes malate in step 7. So the water that's coming in, right, where did it go? Right here. Right? And that's why it's called malate now. And then in the very last reaction of the citric acid cycle, reaction 8, another oxidation occurs where malate encounters malate dehydrogenase, right? And another NAD+. It oxidizes malate, becoming oxaloacetate. And then oxaloacetate moves on to become citrate again, to continue the cycle, to start the cycle over again, right? But the important thing here is we have another NADH that's going to the electron transport chain, right? So notice the difference here, right, between succinate dehydrogenase and malate dehydrogenase, right? Malate dehydrogenase is producing again an NADH that's going to be transferred to complex 1 of the electron transport chain. But succinate dehydrogenase is actually a part of the electron transport chain, so it doesn't have to transfer anything. The FADH that it made is already there. So that is its connected direct connection there. So let's just review that real quick. We have 3 enzymes that are dehydrogenases, right? We had isocitrate dehydrogenase. I'm spelling it wrong. There we go. Isocitrate DH. Then we had alpha ketoglutarate DH. Glutarate DH. And we have now malate dehydrogenase. And the 3 of those all produce what? An NADH that goes to the electron transport chain. And then we had one more dehydrogenase, which was succinate dehydrogenase. And that produced an FADH that was already a part of the electron transport chain. So that's why this is the direct connection to the ETC. Right? Whereas the others up here, the 3 others, were indirect. Make sure you know that. So overall, what's happening here? We can say then that we start out in the citric acid cycle with the acetyl coenzyme A, 3 NAD pluses and an FAD, some GDP, some inorganic phosphate, and some water. And in the end, we ended up with 3 NADHs, an FADH2, and a GTP, and 2 carbon dioxides. Right? So let's count those all up. Coming from one glucose, what do we have? From glycolysis, we get 2 ATPs and 2 NADHs. Remember from pyruvate dehydrogenase, we oxidize pyruvate, and we get 2 NADHs. And then in the citric acid cycle, right, with 2 acetyl-CoAs, we actually get 2 ADPs and an NADH, 6 NADHs, and 2 FADH2s, right? So the total ATPs we get by the end of glycolysis is 4 ATPs, 10 NADHs, and 2 FADH2s. So that's a pretty good energy yield, right? It's a pretty good energy yield. Turns out you get more energy yield actually from a single molecule of fat or palmitic acid, but sadly we won't be able to dive deep into that this semester since this is our last lecture. So now before we can talk about the electron transport chain itself, we've got to talk about the mitochondria, right? The powerhouse of the cell, as you learned in biology class, right? It is the site of the electron transport, and it's also the site of oxidative phosphorylation, right? And so let's just take a look at the organelle itself and where things are, so you can kind of orientate yourself, because the position of these enzymes and these complexes of these protein complexes, right, are very important when it comes to the electron transport chain itself. Okay, and also the citric acid cycle, right? So on the outside of the mitochondria, we have the outer membrane, right? And it is freely permeable to small molecules and ions. Then on the inside of the mitochondria, we have the inner membrane, okay? It's impermeable to most small molecules and ions, including protons, right? And it turns out that in the inner membrane is where the first, or not the first, but the 4 complexes of the electron transport chain are actually located, right? ATP synthase and ATP-ADP translocase are also there, and other membrane transporters are there too. And then in the inside of the mitochondria, right, we have the matrix of the mitochondria, and that's where all the previous enzymes we were talking about are, like pyruvate dehydrogenase complex, citric acid cycle enzymes are there. The fatty acid beta oxidation enzymes, which sadly we won't be able to talk about much because we don't have time in the class left, and also the amino acid oxidation enzymes, right? DNA and ribosomes are also in there too, with many other enzymes, ions, right, and other soluble metabolic intermediates, okay? But just know and remember that the matrix is where the citric acid cycle enzymes are, and pyruvate dehydrogenase is, right? Whereas in the inner membrane is where we find our electron transport complexes, okay? So just a quick review. The inner membrane is highly folded structure to provide more surface area for membrane proteins, right? And it contains the electron transport chain, proteins, and the ATP synthase. The matrix contains most of the TCA cycle enzymes and the amino acid oxidation and fatty acid oxidation enzymes too, right? So know where all these things are located. Now, onto the electron transport chain. So here we have a cross-section of the mitochondria. And I want you to notice a few things first, right? We have the outer mitochondrial membrane right here, which means that this is the cytosol, where all those other things in the cell are located, like the other organelles, you know, like the ribosomes and the Golgi apparatus and the nucleus, they're all in the cytosol, outside the mitochondrial membrane. Then we have the intermembrane space. We'll get back to that in a second. But then we have the inner mitochondrial membrane. Okay? And then over here, we have the mitochondrial matrix. Now, if you recall, the enzymes of the citric acid cycle and pyruvate dehydrogenase, they were all in the mitochondrial matrix, right? The TCA enzymes and the pyruvate dehydrogenase enzyme, right, complex, they were all in the mitochondrial matrix, right? And both of these guys produced NADH, right, when it encountered their respective substrates, okay? Well, those NADHs, if you recall from those steps previous to the electron transport chain, they go into the electron transport chain via the matrix here into complex 1 of the electron transport chain, right? The NADH transfers electrons, right, thereby getting oxidized into NAD plus into transport 1, complex 1 of the electron transport chain, right? So the thing I want you to notice here is that here in the intermembrane space, there's a lot of protons, okay? But here in the mitochondrial matrix, there's not a lot of protons, right? Not a lot of protons, right? And by protons, I mean H pluses, okay? And what happens is that when NADH goes and reduces complex 1, right, or transfers its electrons to complex 1, this pushes protons from the mitochondrial matrix into the mitochondrial, the inner membrane space of the mitochondria, right? The electron actually ends up going into a molecule called coenzyme Q, right there, right? And coenzyme Q takes these electrons from NADH, right, straight to complex 3. Okay, straight to complex 3. We'll go to complex 2, which is right here. We'll talk about complex 2 in just a second. But complex 1 to complex 3, the transferring electron carrier here is coenzyme Q, right? Coenzyme Q transfers its electrons to complex 3, right? And it does this by transferring it to a molecule called cytochrome C. And when that happens, more protons from the mitochondrial matrix are being pushed into the intermembrane space, right? So we're getting this huge accumulation of protons here, and the concentration of protons in the intermembrane space, in the mitochondrial matrix, is very low as compared. So cytochrome C takes those electrons from complex 3 over here to complex 4. Complex 4 is known as cytochrome C oxidase, right? And when it transfers those electrons to complex 4, cytochrome C oxidase, those electrons get transferred to oxygen. This is called so oxygen is called the terminal electron acceptor. Right? Terminal electron acceptor. Right? Because in complex 4, or cytochrome C oxidase, it takes the electrons from cytochrome C and puts it onto oxygen, making a water. Right? So the oxygen that you breathe in every day, right? When you inhale, right? The purpose of that oxygen is to be the terminal electron acceptor here in the electron transport chain. Because if there was no oxygen there to accept these electrons, there would be no place for the electrons to go from NADH and FADH2, right? And then, once that occurs, more protons from the mitochondrial matrix are being pushed up into the intermembrane space, right? And there's a purpose for this, and we'll talk about that in a second, right? But just as a quick review of the reactions that are occurring, right, in complex 1, we had NADH, right, went to coenzyme Q, and then it was oxidized and reduced coenzyme Q to be reduced, right? So the electrons are now on coenzyme Q. And then from there, remember we're going to skip complex 2 for a second. Coenzyme Q transfers those electrons in complex 3 to cytochrome C, and then cytochrome C transfers those electrons over to complex 4, cytochrome C oxidase, where the electrons come and reduce the oxygen to water, and that's why oxygen is known as the terminal electron acceptor, right? All the while, protons are being pushed up through those different complexes, right, into the intermembrane space from the mitochondrial matrix. Now let's look at complex 2 now, which is also known as, remember what is it called? Succinate dehydrogenase, right? Complex 2 takes its FADH2, right, and it also transfers to coenzyme Q some electrons, making it become FAD, and those coenzyme Qs, those reduced coenzyme Qs go straight to complex 3, right? And so the electron transport chain is getting electrons directly from the TCA cycle here, right? When it comes to complex 2, or succinate dehydrogenase. Whereas with NADH, it has to go through all those cycles, and then the NADH gets transferred to complex 1, where it gets oxidized, right? That's the difference. Succinate dehydrogenase, which has the covalently bonded FADH2 in it, is actually a part of the electron transport chain, and that's why we call it the direct connection to the electron transport chain for the TCA cycle, right? FADH2 plus coenzyme Q makes a reduced coenzyme Q. So the reason why these electrons are being transferred from complex to complex, right, is so that it can facilitate the pushing of protons into the intermembrane space from the mitochondrial matrix. The only complex that doesn't do that is complex 2, or succinate dehydrogenase, which you can see why it's only on this side, the matrix side of the intermembrane, whereas the other 3 complexes, they span, right? They span the entire membrane space, intermembrane space, so that it can get protons from one side to the other. But that's okay though, because complex 2 can still contributes electrons to the terminal electron acceptor, oxygen, right? So if you remember back in a previous lecture, we were talking about how hypertonic, hypotonic, and isotonic solutions behave. And when you have a solution which the concentration on the inside of a membrane of a particular solute is lower than the concentration on the outside of the membrane, right? So the concentration on the outside of the membrane is higher than the concentration on the inside of the membrane. In an attempt to become isotonic, the hypertonic solution will try to push its way through the membrane to get to the inside, right, to the hypotonic solution to even out the concentration. So you have an isotonic solution where the solution is the same concentration on both sides, right, of the membrane. Well, that is happening here with protons. We push all these protons up here into the intermembrane space so that it wants to go back through, right, to get back to the matrix, to get more, to get evened out, to get the same concentration on both ends. And it can't just push through, because the intermitochondrial membrane is actually impervious to small ions. So it has to go through a special enzyme called ATP synthase. And when it goes through ATP synthase, it pushes through ATP synthase, making it move, right? We call it a molecular motor, making it move. And in that process, it makes a whole bunch of ATPs. So literally, protons from the intermembrane space are being funneled, right, into ATP synthase to drive a motor to make a whole bunch of ATPs in the mitochondrial matrix, right? While still pushing in. All the while, all these protons are still being pushed out back into the intermembrane space to continue this cycle of making a whole bunch of ATP with ATP synthase. So let's take a closer look at ATP synthase now. Everything I described to you in the previous slide is known as the chemiosmotic model. So, in the chemiosmotic model, each complex in the electron transport chain, that's 1, 3, and 4, act as a proton pump, pushing protons from the oxidation of NADH and FADH2 out of the matrix into the intermembrane space, right? And as I was saying before, it increases the proton concentration, which lowers the pH, and causes this word here, this phrase is what I want you to remember, an electrochemical gradient. To equalize that pH and a charge that's between the intermembrane space and the matrix, the protons are then returned to the matrix, right, by passing through a protein complex called ATP synthase, right? And when that happens, a process called oxidative phosphorylation occurs. Because if you take a look at the picture over here, which I apologize, it's upside down now, because now we have the matrix up here, and over here we have the intermembrane space, right? And here we have the inner membrane, right? So it's upside down now, but the protons are being funneled back into the matrix up here, right? And as it does that, it rotates this turbine, right? And when it's rotating that turbine, as it rotates that turbine and pops a proton back out into the matrix, it also turns this complex here on ATP synthase, right? Which takes in ADPs and inorganic phosphates and pumps back out ATPs, a bunch of them, right? So literally the reaction here is we have ADP plus inorganic phosphate plus energy from the protons turning the turbine, right, with the enzyme ATP synthase to make ATP. And that's how ATP synthase works. It's quite the molecular model. And I know, I'm sorry, this is a little bit confusing because in the previous slide, right, ATP synthase was pointed downwards here, right, with the matrix at the bottom, right? But in the next slide, I needed to have the words upright. And so now the matrix is up here and the intermembrane space is down here, right? But it doesn't matter because the mitochondria is the three-dimensional structure and it can be in any kind of orientation, right? So remember, protons pass through from the intermembrane space, right, trying to get back into the matrix because it wants to get there to equalize that electrochemical gradient and the pH and the charge between the two spots, right? And as it does that, it spins this turbine here, pumping out a proton, kind of like a waterfall, right? And when it does that, it rotates this complex up here, right, of ATP synthase, which has a special name, but I don't want you to remember that, so we're not going to talk about it. And as it rotates it, right, it brings in ADP, and then it's saying diphosphate, and an inorganic phosphate, and pumps out ATP. So let's talk a little bit about how much ATP we get from each electron transfer molecule. So any NADH, when it enters complex 1, right, the energy released from its oxidation can synthesize 2.5 ATPs, right? For every one NADH, you get 2.5 ATPs, right? For FADH2, though, from succinate dehydrogenase or complex 2, for every one of those, you can get out 1.5 ATPs, right? So let's put that all together now and calculate how much ATPs we get from a single molecule of glucose, okay? Remember we talked about this before in a previous slide, but from glycolysis we get 2 NADHs, right? And we get 7 ATPs, right? And then in pyruvate dehydrogenase, which is this line right here, we get 2 more NADHs and 5 ATPs, okay? Then in the citric acid cycle, right, this has every enzyme that produces something. Isocitrate dehydrogenase gives us 2 NADHs plus 5 ATPs. Alpha ketoglutarate gives us 2 NADHs and another 5 ATPs. Then we have direct transfers of phosphates from GDP to make ATPs. That's 2 of those. We get 2 more FADHs and 3 more ATPs from succinate dehydrogenase. We get 2 NADHs and 5 ATPs from malate dehydrogenase, right? So in summary, after all that is done in the citric acid cycle, we get 20 ATPs, right? Add that to the 12 up here, right? And we get a total of 32 ATPs from a single glucose molecule, right? From a single glucose molecule. And that's how we sustain functions in life and cellular respiration. Oh, by the way, this is called cellular respiration. I don't think I ever said that. Cellular respiration. That's how we sustain cellular respiration, right? In our bodies. In all animal bodies, even plant bodies, right? Plant bodies. In animal bodies. In our cells, right? So that's called cellular respiration. You get 32 ATPs, okay? 32 ATPs. And that, I believe, ends this lecture.